Hello, my name is Anton Korobeynikov, and today I'm presenting our joint work on BinSPreader. BinSPreader is a tool that might be used to refine the binning for the fuller MAG reconstruction. So first of all, what are MAGs? MAG stands for the metagenome assembled genomes. MAGs are usually derived from the complete metagenome assemblies via the dedicated binning process. Often they are agglomeration of genome assemblies of closely related species or strains of the single species, and therefore they are often incomplete, they are often contaminated, but usually it's usually it's the final result of a metagenome assembly, and therefore MAGs are quite different from the usual isolate assemblies. The conventional workflow to derive a MAG is to start with the normal sequencing reads, then go through the assembly using the metagenomic assembler like MegaHit or Metaspades or IDBUD or whatever, or Metafly for the long reads. Then the resulting contigs are clustered together using for example, coverage or abundance estimation or different sequence content, for example, the tetranucleotide frequency profile. And this clustering process is called binning, and the resulting genomic bins are further refined in order to obtain the fuller MAGs. There are lots of different tools for the binning developed over last few years. But the real workflow for the binning and for the binning refinement could be quite complex. Here is an example of the binning and binning refining from the MetaRap. It involves running multiple binners together and then kind of join or take the consensus of the binning assignment via the dedicated bin refinement procedure. What is the problem? What are the problems with the MAGs and why they are often incomplete? In many cases, some contigs from the whole metagenome assembly are missed from the relevant bins. So usually they're short contigs because in order to reliably estimate the sequence content, the conventional binners only work on the contigs that are longer than, say, a couple of kilobases. The conservative genes or genes that undergo horizontal gene transfer or something similar often belong to multiple species, right? So they represent the so-called interspecies repeats in the assembly graphs, and if these repeats are unresolved, then they are likely missed from all relevant bins. So sometimes they are missed from all the bins, sometimes they only turn out into just few bins. Also, strain variants, the uncollapsed strain variants also causes some fragmentation of the assembly, and therefore the context representing the strain variation is also likely missed from the relevant bins. There are different tools that might be used to refine the existing binners, and there are different approaches. Some of them are, I would say, even orthogonal to each other. The first couple tools called DASTool and MetaRap and among the others, use the consensus from multiple binners in order to increase the purity and possibly even increase the completeness of the resulting bins. Recently developed Pinnacle use paired entries in order to refine the binning and increase the completeness and join several bins together, and GraphBin and GraphBin2 family of tools use the scaffold graph in order to refine the binning. So BinSPreader is yet another binning refining tool. However, what is, I would say, novel here is that BinSPreader is using assembly graph for the binning refining. Also, we try to include few features into BinSPreader thinking of possible usage of it and therefore simplify the downstream analysis of its of results. First of all, BinSPreader is capable to fix possible binning errors. BinSPreader could possibly infer multiple, even overlapping bins for the contigs. Oftentimes, reads belonging to the single mags are reassembled in order to improve the contiguity of the mag. And BinSPreader also helps with the dedicated read splitting procedure. More on this later on. Also, BinSPreader is able to calculate some kind of the distance between bins according to their overlap on the assembly graph, and this thing could be used. This similarity could be used to join multiple bins together if they're split. BinSPreader uses as the input several flavors of assembly graph in standard GFA format, for example, from Metafly, MegaHit, Metaspades, and some other tools. What is different in BinSPreader? First of all, BinSPreader uses the assembly graph, so unities or edges of the assembly graph in order to produce the refining. So essentially, BinSPreader does the refining procedure on the assembly graph and not on the scaffolds. So why do we need to break the longer contiguous scaffolds down to the edges? Here's an example. Consider the partially unresolved repeat in the assembly graph consisting on 3 different contigs. Like the blue path is a single contig and 2 red edges are separate contigs. And consider that the blue contigs belong to the one bin and red ones belong to another bin. If we take the scaffold graph, we try to do the refining using the connectivity of scaffolds together, trying to assign the closely located scaffolds to the single bin, then we might end with the inconsistent or even incomplete refinement. But if we use the edge adjacency on the assembly graph, and we might be able to detect that the middle edge in the middle is actually a repetitive one and assign it to the multiple bins. BinSPreader uses the so-called sparse, noisy, multi-label propagation framework, similar to what is used for the community detection in the social networks and similar graph clustering approaches. So essentially, the whole idea is to attach to each edge its soft bin assignment vector containing the weights or rather, I would say probabilities whether the particular edge belongs to the particular bin. And then the whole binning or binning assignment of the graph could be represented as the soft bin matrix. And now, if we have the initial binning matrix with the ones if the particular edge belongs to the particular bin, then our task is to find the graph soft bin assignment matrix somehow close to the initial binning. But what is close and how to find? BinSPreader operates within the so-called trivialization framework, and the whole idea is to find the proper balance between the smoothness of how the bin assignment changes when we work over the graph. So certainly we expect that close edges will have a similar bin assignment. And on the other hand, we want to keep as much of the initial binning as possible. So all this is grouped together, and therefore we end with this kind of the regularization, with the minimization problem with possible additional regularization. What is interesting here is that we could use the regulation parameters to do or to tune the overall refining procedure. First of all, if we took the regulation parameters as infinity for all binned edges, we essentially do the binning propagation. So we never change the binning assignment for the already binned edges. But if we take the regulation parameters less than infinity, then the labels might be possible changed. So this corresponds to the essentially binning correction. Also, we never trust bin assignment of the possible repetitive edges because of the partially unresolved repeats. So if we detect that edge belongs to the multiple scaffolds, then we never trust the initial binning. Short edges are likely repetitive, and therefore, by default, we put the regulation parameter proportional to the length of this edge. So we likely correct the binning of the short edges, but the longer ones are usually correctly binned and represent the unique sequences, and therefore they're much less likely change their binning. Then we have the binning assignment for the whole assembly graph. We come back to the scaffolds and assign the bins for the separate scaffolds via maximum likelihood. And again, here we have the choice via maximum likelihood and having just a single bin assignment. And also we could use the multiple bin assignment, for example, take all bins that together explain up to 95% of the likelihood. As I said, BinSPreader is able to align reads down to the refined binned assembly graph. So it split the reads into separate bins according to the bin assignment, so the reads are ready for the possible reassembly. What is also interesting here is the possible overlap between bins. Like close species have likely overlapping bins, and it might be important to detect this properly. So the question is how to measure this. Unfortunately, Jacquard similarity won't work because we have self-bin assignments, and what we use here is the special probability Jacquard index recently proposed by Moulton and colleagues. And this similarity measure could be even used to merge bins that are split in the assembly graph according to the overlap. So the whole idea is that we treat each bin as probability distribution on all scaffolds. Then we calculate this probability Jacquard similarity measure. And good things here is that it's scale invariant on both arguments, so it's not lower than ordinary Jacquard distance for discrete uniform distributions, and also if we take this similarity into this similarity, then it's a proper metric. Let me show some results. So I will use three quite common benchmarking metagenomic mock datasets, namely SIND containing 64 bacterial and archaeal species, EMBARK containing 23 bacterial and 3 archaeal species, and BMOC containing just 11 bacterial species, but 5 of them are closely related, like 3 and 2 strains correspondingly. Here you can see the hierarchical clustering trees built from the bin distance. As you could see here, and I colored them according to the reference, so the bins belonging to the single reference, they are clustered quite well together. The ones that are overlapping actually belong to the multiple strains of the same species, so we are also good here. If we take into the MAC completeness, as you can see, as compared to the GraphBin2 and MetaBud, which corresponds to the no refining at all, for EMBARK, BinSPreader is able to derive almost all genomes here. The situation with BMOC is slightly different because it contains lots of similar species, but still we have quite good results here. If we take into the completeness and purity figures, they are also quite good here. So BinSPreader is able to derive less bins as compared to the GraphBin2 and MetaBud, but these bins are more complete and more pure. The similar situation is on BMOC 12, so the results are quite consistent, I would say. In the beginning, I told that many conservative genes, for example, ribosomal genes, are missed from the MAGs, and it's quite a problematic thing. So in order to check how the binning refinement works here, what we did is that we checked how many, like the proportional of MAGs with 16S context before and after refining. And you could see that consistently on all datasets, binning refinement certainly reconstruct, is able to recover lots of 16x context, therefore enabling downstream analysis. BinSPreader is quite universal, so it could use different sources of bin linkage information, including paired end reads, high-C data, long reads. Also, it could refine the whole assembly graph or keep some edges unbinned in case when the binning is not complete. There are some few final touches that we hope to be able to finish by the time you are seeing this talk. But in any case, contact us via paid support email for the early preview version. I would like to thank our colleagues for the overall support of this work, and this work was supported by the Russian Science Foundation, and I will take any questions. Thank you.